Hello Med1, welcome to part 2 of the 4-part series of videos on the cytoskeleton. In this video, we'll be focusing on microfilaments. So taking a look at the first learning objective, let's make sure I have my pen working here, okay. So taking a look at the first learning objective, okay, there we go. We're going to be looking at where within the cell our microfilaments are found, what their functions are, and the types of proteins that bind to them to help them perform their functions. We'll also look at how these microfilaments build up and break down or polymerize and depolymerize, and basically why you need to know about that. Okay, so microfilaments are found in just about all cell types. In non-muscle cells, some examples include epithelial cells, including in the microvilli of epithelial cells. So let's look at the graphic on the right. We see on the far side of it, we see them around the cell, basically around the periphery of these epithelial cells, helping to maintain the shape of the cells. The adhesion belt, we'll be covering adhesion later on during foundation, so I'll leave that for now, and also in the microvilli. So you might know that all cells have microvilli, but on intestinal epithelial cells in particular, and some cells in the kidneys too, these microvilli are much longer and they're important for absorption of nutrients. So again, microfilaments are found there to help maintain their shape. In migrating cells, cells that move, an example, fibroblasts. Fibroblasts are a major cell type in connective tissue. White blood cells is another example. White blood cells are, well, as the name suggests, found in the bloodstream. But I'm not referring to movement of these cells within the bloodstream, since bulk flow of the fluid is going to move them through the blood vessels. I'm referring to when a white blood cell needs to move from the bloodstream into the nearby tissue to fight an infection there, for example. They need a mechanism to move into the tissue, which is solid, not fluid like blood, and towards a specific place within that tissue where an infection might have set up. Think of downhill skiing. If you're not in a race, just skiing for pleasure, to get down the hill, you almost don't need to do any work, right? Gravity is going to take you down. But if you need to get from the slopes into the chalet or the ski house or whatever, to go to the change rooms, there needs to be some mechanism, a different mechanism, to take that walk from the slopes into that place. Another type of non-muscle cell is in auditory hair cells. So these are cells that are found in the inner ear that contain actin. And in particular, these cells have stereocilia. Stereocilia, basically small hair-like projections, and they refer to as being hair-like because they're thin, very, very thin. They're important for helping us, they're, in a part of the inner ear, they're important for helping us maintain our balance and even for detecting certain types of sounds. So these stereocilia are different from what I'm going to call regular cilia that we're going to talk about in a subsequent video. But actin are important for maintaining the shape of stereocilia. Let's go over again onto the right side in the middle picture. So we see some examples of how microfilaments are arranged in migrating cells. So again, around the cortex, always maintaining cell shape. But here we see them forming a lamellipodium, which is singular. The plural is lamellipodia. And basically, see how the actin is arranged. Basically, that it has helped the cell deform to form what we call a false foot. More about that later. And in the other picture here, we see filopodia. Filopodia, basically these small finger-like projections, where again, we see actin is arranged within them, helping them maintain the shape. Both the filopodia and lamellipodia help cells to migrate. In the lower part of the picture, we see stress fibers, which we're not going to talk about in this video, but we also see the contractile ring. So any cell undergoing mitosis has to eventually form this contractile ring, which is what is going to allow the production of two daughter cells. So let's move on. We also find microfilaments in muscle cells, and muscle cells are where actin is essential for contraction. We'll talk a bit more about that later. So let's go now to talk about another aspect. So I say here, actin is a central component of microfilaments. And in the first video, I said we tend to use the term actin and microfilaments interchangeably, and yes, we do. But I put the title in this way just to tell you that microfilaments, there are other accessory proteins found in different cell types depending on the function, but actin is the constant and major component of microfilaments, which is why we tend to use the terms interchangeably. Okay, so I mentioned before that actin is found in all cell types, but there are different isoforms. And isoforms meaning, when you look in the textbook, you'll see reference being made to alpha actin and beta and gamma and what have you. And yes, these all exist. So I want to say that even though we tend to just say actin in the singular, it's not one protein, it's a family of proteins. That being said, that's all you need to know. You're not required to know the different isoforms, where they're found, what they do, any of that. Okay, so this is just FYI. In the picture on the right-hand side, we're looking at a fluorescence microscopy image of a fibroblast, actually, a fibroblast cell in which the actin is stained green. And notice how we're seeing the actin concentrated mainly around the periphery of the cell. So again, helping the cell maintain its shape. If you take a close look, you see some light staining in the inner parts, in other parts of the cells, for example, going across the nucleus here. So there is some research saying that you can actually find some actin within the nucleus, but if you're ever asked a question on an exam about where actin is predominant, your answer is always going to be in the cytosol. Okay, so let's talk about how we get our actin filaments. When we talk about actin, we are talking about F-actin most often. F-actin referring to filamentous actin, okay? But in order for us to get to that filament, we have to start with monomers, okay? G-actin monomers. So I'm going to backtrack a bit to the first point here, and then I'll tell you a little bit more about that. Our actin, our F-actin, is, we can use a couple of words to describe it. One of those words is polarized. So what do we mean by that? I'm going to just draw F-actin as a simple straight line. Now, one end is referred to as the plus end, and another end is referred to as the minus end. Why? Because they're different. They are structurally, chemically a little bit different from each other, and that is what we refer to as being polarized, when the two ends are different. Actin may also be described as being dynamic. So when you hear the term dynamic, you think of something that's changing or moving, and those are perfect words to describe actin. It's not just a static one filament, one straight line that's this long and nothing else will happen to it. Actin can get longer, the filament can get longer, it can get shorter, it can change depending on its function. So that's why we describe it as being dynamic. So let's go back now. So we know we have a bunch of G-actin to make F-actin. G, by the way, stands for globular. So let's move to the picture on the right-hand side, and we see our G-actin monomers sort of at this globular shape. We have part of a filament here. Okay, this is our filament. This is the plus end of that filament, and this is the minus end of the filament. And here's another globular actin, another G-actin, and that is wanting to join the party, to become a part of the filament. So note here that the actin at the leading edge, or the plus end of the filament, has ATP bound to it, little red ball. And the one that's coming in also has ATP bound, and that's a requirement. So in order for actin to polymerize, ATP is required. Hence, in the third point, moving back over here, we see G-actin needs ATP for polymerization to occur. And when the new G-actin adds to the filament, we have hydrolysis of ATP, meaning that the ATP is basically broken down to form ADP, adenosine diphosphate, and inorganic phosphate. The picture at the bottom, there are a few words in the boxes there, and I'll leave you to read that. The last point that I'll just mention is that polymerization tends to occur preferentially from the plus end. Depolymerization occurs preferentially from the minus end. And we're going to come back to this whole polymerization and depolymerization in a bit. But in some cells, we don't necessarily want actin to be polymerizing and depolymerizing. We don't want its length to be changing. So think about your skeletal muscles, the muscles in your arms, the muscles in your legs that are important for you to do work, whether lifting weights in the gym or picking up something. You don't want to look at your muscles and see them getting longer or shorter, as the case may be over time. The actin in your skeletal muscles needs to maintain a consistent length. And to do that, because actin is so dynamic, it wouldn't naturally happen. So to do that, we're going to put what we call a capping protein at the plus end and a capping protein at the minus end. And what these proteins will do is prevent the filament from shortening from the minus end or lengthening, for that matter, from the plus end, maintaining a consistent length. Okay, so let's talk about some functions of actin. Major functions: structural, as I've been discussing so far, and motility, as I've also mentioned. First off, helping to maintain cell shape. And I put here a note, cross-reference spectrin. So let's take a look at the picture on the right, the upper picture. There are a bunch of labels there, but I want you to focus just on the two that are in the red circles, actin and spectrin. What you're looking at here, so all this blue part, you're looking at a part, just a section of a plasma membrane, okay, the membrane of a cell. And just imagine you're in a room and you're looking up at the roof. So this is the view that we have here now, the inner side of the roof, or the cytosolic side of the roof, quote unquote roof, or the plasma membrane of the cell. So note how our spectrin and actin are sort of cross-linked all along the inner part of the membrane. And that is how spectrin, which is an actin binding protein, helps to maintain the shape of the cell. Now, you find this in just about all cell types, but let's use the example of a red blood cell, which is shown here in the picture, an erythrocyte. Erythrocytes typically have a biconcave disc shape. And that shape is important for helping them to fit through our blood vessels, especially the very tiny capillaries. The actin has to be able to be pliable. The membrane has to be able to squeeze through. Imagine yourself trying to fit into the skinny jeans. Sometimes you find yourself wiggling this way and that. Well, the red blood cells, the erythrocytes, need to be able to wiggle this way and that to be able to get through the capillaries. And you know what? These cells do so quite efficiently. The spectrin bound to the actin is important for maintaining that shape. Now, when you read the textbook, there are these boxes with these little clinical pearls. And in one of those, you're going to see a disease mentioned called hereditary spherocytosis. In that disease, there is a defect in the spectrin, so it's not able to cross-link the actin effectively, as you see in the picture here. The effect of that is that the red blood cells lose their biconcave disc shape. They become more spherical. And believe it or not, when they're spherical, they're not able to fit through our tiny capillaries as effectively. So what may happen is they take longer to pass through, or they may even not fit through. They end up being blocked. And so patients with this disease, you might find them having hypoxia because those red blood cells are not able to carry oxygen to the downstream tissues. These cells would eventually be destroyed, so those patients might eventually have anemia also. Okay, so some other examples of providing structural support to plasma membranes. I put in red here the protein dystrophin. And you'll find that mentioned both in the reading guide and in the textbook. So dystrophin is another protein. It's not shown in the picture here, but it's also important for cross-linking actin. And you see the effect most prominently in muscle cells. So to maintain the integrity of the membrane of the muscle cells, dystrophin is one of the key proteins. So much so that if dystrophin is affected, you'll find that those patients have issues with their movement. In the textbook, again, another one of these clinical boxes, it talks about different types of muscular dystrophies, like Duchenne's and Becker muscular dystrophy. In those patients, because of the defect in dystrophin, over time, they become less mobile. Eventually, they end up in a wheelchair, and their life expectancy is significantly shorter than a person without this type of defect. Another example of where the actin is important for maintaining cell shape is in microvilli, and I've talked about that before. So I'll leave you on your own to read through what is in these bubbles on this slide right here to be able to appreciate that even better. Let's move on now to another function of actin, restricting diffusion of organelles. So in the picture on the right, we're seeing a cell that has gone through mitosis, telophase, and then cytokinesis is happening here, which is where we need to form that contractile ring, and the ring is eventually going to pinch this structure into two so that we can have two daughter cells. Even within, whether in a mitotic cell or regular type of cell, so let's go back to the text on the right now. We see one of the functions is restricting diffusion of organelles. So what are we talking about there? Remember, actin is concentrated mainly around the periphery of the cell, right, around the cell cortex. So within the cell, we have all our various organelles, our nucleus, ER, what have you. And those organelles have certain degrees of freedom. They can move a little bit, but we don't want them moving so much that they end up bumping into the plasma membrane. So our network of actin there helps protect those organelles. Believe it or not, imagine your nucleus going bumping into things. We don't want that. It restricts diffusion in that way. It also provides an anchor for cell adhesion molecules. So we're going to see an example of that coming up in the next slide. And we're going to talk about adhesion later on. So I'll leave that there for now. And then in the last point here, contraction. So contraction, I put here cross-reference myosin because myosin is important. Is an important component along with actin for contraction in muscle cells, just as important as actin. In non-muscle cells, however, so that's where the picture on the right comes in. I sort of jumped the gun a little bit there. But as I was mentioning, in telophase of mitosis, you find this contractile ring forming, and once it eventually pinches this into two, we get the two daughter cells. And then I put other. So other can be any number of things, but one example I'll give you is wound healing. In order for wounds to heal, we need to have that actomyosin contractile ring forming. If you've ever gotten a cut, a small cut, nothing that needs stitches, and look at it over days as it begins to heal, you'll see the edges almost sort of a tugging. You can see it on the skin, and you may actually feel it, feeling kind of the skin in that area is a little bit tight. Well, the actomyosin contractile ring has formed, and that's a part, just one part of a larger mechanism to close that wound. So let's move on to talk a little bit about the cell migration or cell crawling. Now, this is necessary for various types of events. One of them being embryogenesis. So I'm not sure if everyone knows what this is, just in case not. Genesis refers to the beginning, an embryo. Well, you have an embryo being formed subsequent to fertilization of an egg by a sperm. So during embryogenesis, we'll need to have cells moving from one layer to another, and that movement is going to require action by actin. Another example is for movement of axons in response to growth factors. So we're talking about chemotaxis there. Chemotaxis is where, well, in this case, we have a part of a nerve cell, also known as a neuron, responding to growth factors by growing towards them. So the growth factors are basically chemical molecules, and the word taxis, T-X-I-S, refers to movement. So this is what is being referred to here. Then movement of white blood cells toward a site of infection. I talked about that a bit on a previous slide. And last but not least, phagocytosis, which is when a cell needs to engulf something that needs to be destroyed, such as an invading bacteria or a virus. So for phagocytosis to occur, we need to have actin playing a part. And then the second point that we have here, it says the leading edge will temporarily attach to the matrix. So for discussion of that, let's look at the picture on the right-hand side. So look at our starting point and look at our finish line. That cell basically needs to move from the start to the finish. Now that cell started out, most cells are roughly spherical, most of them, not all. But notice the shape of the cell here. It's starting to look like a car almost, right? Well, part of that is due to actin and the dynamic nature of actin. Actin has been polymerizing in this direction. So I'm going to call this the plus end and depolymerizing from the minus end. So notice here that there are these sort of a false feet where the cell is attached to our extracellular matrix, which is referred to as a substrate in this picture. Actin is important there. Then you have this protrusion at the front, and look at what eventually happens. That protrusion ends up also binding to the substrate right here. Actin is important for that too. While that leading edge, that protrusion, is binding to the surface, look at what's happening in the next frame, where that false leg is lifting up from the substrate. So that is because actin is depolymerizing from the lagging or the minus end within the cell. So this process of forming a false foot, you have the leading edge that attaches to the matrix, the part at the back, the lagging edge, the minus end, detaching, All of that is due to actin within the cell, polymerizing and depolymerizing as we go along until that cell finally reaches the finish line. Yay! So let's talk a little bit more about that, about the cell crawling. Now, instead of actin simply polymerizing in a straight line, it often branches to help the cell migrate. So let's look at the graphic first, and then we'll come back to the words. Cells are going to start out, let's look down here on the left side. So this is actin within a cell, within the cytosol of a cell, and this cell likely started out with a simple, roughly spherical shape like that. Eventually, the shape changed, the cell deformed because our actin has been branching, and eventually we end up with the cell having a shape like this, as I'm drawing here in red. Okay, I'm cutting off the actin a bit, but I just want to emphasize what is happening here. So instead of just instead of just the polymerization happening only in a straight line, perhaps as we're seeing here, it's forming all of these branches along the way. And we're going to talk about the why in a second, but let's talk about the how first. Notice that the various branches start at this green node, okay? And that green node is basically these proteins called ARP2 and ARP3. ARP refers to actin-related protein. So ARP is needed for branching, and branching is needed for migration of these cells, right? What we've done here is we've formed a lamellipodium by protruding the plasma membrane as we have here. So that's the first point in words. Actin branches through the activity of ARP2 and ARP3. In the second point, we have this protein called Wiskott-Aldrich syndrome protein. And that is important for stimulating ARP2/3 to nucleate F-actin formation. What do I mean by nucleate? Think of nucleate as to start something, okay? Nucleate, you're thinking about nucleus in the middle of something where the most important thing is. Well, here we're using the word in terms of starting something. So we want to start a new branch with our F-actin. We need this to happen. So again, ARP needs to be stimulated to start a branch. The stimulation of ARP2/3 is the job of the WAS protein. So WS stimulates ARP to start a branch, and the branching is needed for migration. Say that a few times. Okay, there's an inherited disease called Wiskott-Aldrich syndrome. Your patient with this syndrome might might say, Doc, I have this cold and the cold has lasted for weeks. My friends get a cold and the cold is done in a few days to maybe 1 week, but mine is taking that much longer. Well, once you figure out what's going on, you recognize that because his immune system cells have a defect in this WAS protein, that has affected their ability to crawl or to migrate to the site of infection where the virus, for example, may be causing the cold or whatever bug it is. And that is what is going on with these patients. They tend to be susceptible to infections. They don't get over them quickly. In the figure on the right, that's just another representation of what I just discussed. Okay, so let's move on to talking about thin filaments. So this is another in our lineup of synonyms. Actin, microfilaments, thin filaments. Well, we tend to call them thin filaments, particularly when we're talking about muscle cells. Now, thin filaments are composed mainly of F-actin, but not only of F-actin. There are some other proteins, other actin-binding proteins. One of them is tropomyosin. So tropomyosin, notice it in a picture here. Tropomyosin is an actin-binding protein. And then the troponin complex of proteins, troponin I, troponin C, and troponin T. This complex is found in skeletal muscles, like the ones in your arms and legs, and also in the muscle cells of the heart or cardiac cells. They're supremely important. So you're going to learn more about these in the MSK system, but I do want you to know about them right now. So let's talk about a clinical scenario again. When a patient comes in and you suspect that that patient may have suffered a heart attack, one of the things that you're going to do, one of the tests you're going to order is to look for troponin. So you're going to send an order to the lab saying that you want to find out whether or not there are troponins. They're going to take the blood. They're going to separate the cells from the plasma, and they're going to test that fluid, the plasma, to see whether or not they can find troponins. So how is this significant? Remember, our cytoskeleton is within the cell, right? That includes our actin, and that includes these troponins that are bound to actin. So if the lab does a test and finds that these troponins are in the plasma, they're outside the cell, well, that's a significant indicator that the cells, in this case the cardiac cells, must have ruptured to spill these troponins out into the plasma. And there are certain troponins that are found particularly in cardiac cells and nowhere else. That's why it's okay to look for them, and there'd be an indicator that there's some sort of a cell destruction that has been occurring within the cardiac muscle. I'll leave that there for now, but definitely you'll have more on that later. And then if we have thin filaments, that suggests that there's probably a thick filament out there somewhere, and there indeed is, and that is called myosin in the case of muscle cells. So our thick filaments, or myosin, is another actin-binding protein, and it's also described as being a motor protein. Whenever you see the term motor being used, that means energy is involved, usually in the form of ATP. So myosin binds ATP. Now, mind you, I mentioned that actin and myosin can form contractile rings in all different cell types. So you find this in muscle and also in non-muscle cells. And just like with actin, there are different isoforms of myosin, but again, you're not required to be able to distinguish the different isoforms. I've put the electron micrograph below just as to sort of a seed your knowledge, to get you started for when MSK comes. So this is the contractile apparatus of a muscle here. And in this, you see a lot of these repeating units. The myosin stains really darkly, as you're seeing here, okay, because they're thick filaments, and the actin, because it's so thin, it doesn't stain. So you just see this white region here. That's where our actin is. But again, more of this later. I want to introduce you to it now. So in this video, we've talked about where in the cell actin can be found. We talked about different actin-binding proteins, and we talked about different functions of actin. Everything from muscle contraction to motility to maintaining cell shape. And we touched a little bit about how actin polymerizes and depolymerizes, and how that's important, particularly for changing the shape of cells, for cells to move. Join me for the next video. We will talk about another aspect of the cytoskeleton.